Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati, and we were discussing about the electrophoresis. So, in the previous lecture we have discussed about the basics of the electrophoresis and how the electrophoresis is actually separating the different types of molecules and how the technique is being evolved. So, initially the people were using the moving boundary electrophoresis, and then they have overcome the deficiencies in that particular technique by the introduction of the solid support media, for example, the acrylamide or the agarose, and as a result they have developed the zone electrophoresis. So, the gel electrophoresis is one of the form of the zone electrophoresis, and then within that we have discussed about the vertical gel electrophoresis, how to cast the gels, how to perform the gels, and what are the different mechanisms of the stacking of the sample and as well as the other kind of minor details. And at the end of the previous lecture we have also discussed about the how to perform this electrophoresis in the gel. So, in that I have taken you to the my lab and the students have shown you how to cast the gels and run the gels. So, in the today's lecture we are going to discuss about the staining of the gel and as well as how once you got the stained gel, how you can be able to do the image analysis. We have the multiple options in terms of the different types of stain, and we have the advantages as well as the different types of options available with the different stains. So, we have the Comasi Brilliant Blue R250 as a stain, and the Comasi Brilliant Blue R250 is the one of the most popular stain what people are using for the acrylamide gels, and it is non-specific. So, it does not stains the any particular type of protein. For example, it cannot stain the phosphorylated protein or stylated protein, but it will stain all the protein what is present in the gel. But one of the major advantage of the Comasi Brilliant Blue mediated staining is that it is a one step simple staining procedure, and the major disadvantage is that the detection limit is in the range of the microgram per protein band or the microgram per protein present in the lane. Then to improve this, people have developed the colloidal Comasi Brilliant Blue solutions. So, the colloidal Comasi Brilliant Blue solution has the 5 to 10 fold more sensitivity than the normal Comasi Brilliant Blue R250, and added advantage is that you do not need to do these staining steps. Then compared to that, people have come up with the silver staining. The silver staining is even 100 folds more sensitive than to the Comasi Brilliant R250, but the disadvantage is that it is not a simple staining procedure. It requires multiple steps and it requires the different types of reagents, and on the other hand it is also not the mass spectrometry incompatible, which means if you stain the your gel with the silver stain, you cannot use the protein from directly for the mass spectrometry. To overcome this, the people have developed the three more the stains like the Cybro Ruby, Cybro Orange and the Cybro Tangerine, and all these are actually the mass spectrometry compatible dyes, and they are sensitive up to the 1 to 2 nanograms of per protein bands. And for example, the Cybro Orange, it is a orange color dye, and then Cybro Tangerine is also is a very sensitive, and it is also compatible with the mass spectrometry. So, even after the staining, once you got the band, you can be able to use the downstream applications like the mass spectrometry. You can identify the protein with the help of the different proteomics approaches. So, in a typical staining procedure, what you are supposed to do is you have to first take the dye and then you have to prepare the staining solutions, and once you prepare the staining solution, then you incubate the gel into the staining solution, and then you have to perform the de-staining steps, or in some cases the de-staining steps is not required. So, to explain you the staining procedure, we have taken an example of the Comasi Brilliant Blue and how to perform the staining procedure. So, for the staining of the gel with the comestibillion blue, the material what you require is the polyacrylamide gel which contains the protein bands. Then you require a comestibillion blue R250 staining solutions. Then you require a de-staining solutions. So, the de-staining solution which contains 15% methanol, acetic acid 10%, and that all you are going to prepare in the triple distilled water, and you require a plastic or the glass container with the lid, and then you need a shaker. To perform the comestibillion blue mediated staining, first what you have to do is you remove the polyacrylamide gel from the electrophoresis unit and place it in a plastic container with 10 volume of the comestibillion blue R2 staining solutions. You agitate the slowly on a platform shaker for 30 to 60 minutes. Then you discard the staining solution and wash the gel with the triple distilled water. So, that actually is going to remove the excess dye what is being attached to the gel, and then what you can do is you can add the 5 to 10 volume of de-staining solutions, and as a result what will happen is it is actually going to remove the dye from the non-specifically bound places, and that de-staining solution you have to keep the gel in the de-staining solution for 30 to 60 minutes or until you will not going to see the bands, until you will not see the bands, If the color of the de-staining solution is intense blue, you can replace it with the new de-staining solution so that it can be able to utilize, or it can be start removing more and more dye from the non-specifically bound places. And if you do so, let us see, if you stain the gel with the comestibillion blue in the beginning, the gel will be look like as a black film, and this is all because the comestibillion blue is going to bind every places irrespective of whether you have the protein or not, which means it is actually going to bind to the polyacrylamide as well as the proteins, and then but the affinity of the comestibillion blue for the polyacrylamide as well as for the protein is going to be different. So, once you do the de-staining step, so what will happen is the comestibillion blue is going to be removed from all the non-specific polyacrylamide gel, but the place where you have the protein, it is not going to be removed because it has a more affinity for the comestibillion blue. So, as a result you are going to start seeing the protein bands. So, this is the typical pattern of the protein band what you are going to get after the de-staining steps. What you can see is that even between the bands also the dye is being removed by the comestibillion by the de-staining solutions, and this is what you have to achieve simply by going into the de-staining solution. So, after the every round of de-staining solution, you have to observe the gel whether you are getting the bands or not, and accordingly you can optimize that particular step to see that you are whatever you are seeing is actually the adequate, and it is actually going to give you the appearances of all the protein bands present in the lane. Now, once you got this band, once you got this image, what you can see is, and what you can be able to analyze the two things. One is the position of the band, the second is its distance from the origin, and based on this, and apart from that you can also be able to see what is the intensity of this particular band. So, these are the three information you can be able to infer when you will actually going to do the image analysis after the staining procedures. So, image analysis is a very complicated procedure because it requires the understanding of not only the band, the band of the protein band, but also the neighboring backgrounds. So, that is why image analysis requires the some kind of assistance from the softwares which actually is going to integrate and which is actually going to change the image into a binary format, and as a result all these spots are going to be converted into the binary format of 010101 like that, and as a result what you can see is that it actually is going to change the image into a kind of a 01 format, and as a result what you will going to see at the end is, for example, if suppose this is your band, okay. So, what will happen is every spot is going to be converted into 01 approach, which means wherever you do not have the intensity, that will be considered as 0, whereas wherever you have the protein band, it is going to be considered as 1 or 10 or 50, or so that depends on the calibration of the softwares. So, if you can calibrate the software and it will actually going to analyze the your image, it can actually assign a random number to particular intensity, and as a result you can be able to convert this whole image into that particular format, and that actually helps the softwares to understand each and every pixel which is present in the image, and that is how you can be able to identify the position of this band. You can be able to measure the intensity of this band. So, for this purpose we have couple of softwares which can be used to perform this function. So, these are the different softwares what you can use for the polyacrylamide gel analysis. These are VisionWorks or the doc itself, which is from the UVP Incorporations, Total Lab Solution series, which is from the Non-Linear Dynamics, Gel Pro or Image Pro, which is from the Media Cybernetics, NIH Image, which is actually a free softwares which is available from the American funding agency NIH, and then you have Melanie or Image Master, which is from Expasy, and then you have the Image Master, which is from the GE Healthcare. Now, to analyze a image, we have the discrete steps. So, what is there in the step 1? The step 1, you have to first detect the lane positions, the lane in which you are interested to do all the image analysis operations. So, in the first step in the image analysis, you have to identify and mark the lane positions, which means if you have a lane, you have to first mark the lane that I want to analyze this particular protein land. There are different ways to do this. For gel with the well defined bands, automatic lane detection algorithm can be able to detect the lane. If the bands are smiling or not straight, then a manual positioning and identifying lane is the best. There is a necessary consideration while doing manual positioning. The bracket used should be large enough to cover the whole lane, but it should not be wide enough to include the lane of the other lane. So, when you are doing the first step you have to detect the lane in which you are interested to do the measurements. This has to be very precise, so that it should only detect the lane 1, it should not going to take up the bands from the neighboring lane, which means if you have a 2 lanes side by side, you can just select the lane what you are interested to measure, but it should not be so big that you actually going to take up the band from the neighboring lane as well. So, once you are done the detection of the lane position, then you have to go for the step 2. In the step 2, you have to do the bands in the band which you are interested to do the position, to know the position of that particular band as well as to know the intensity of the band, which means within the lane you can have the multiple bands, and then within this lane you can also select the band which you are interested to measure or which you are interested to analyze. Once the lane is defined, your protein band in the lane can be defined by systematically scanning the lane profile and identify the region of the local maxima as a band. Then the step 3 is that you have to do the background subtraction. So, once you select the band, you are not going to select the band, but you are also going to select the neighboring background. So, that background has to be subtracted from your protein band, so that you are going to get the absolute intensity of that particular band. So, the background plays an important role in identifying the protein band as well as measuring the protein band intensity. Background of a gel picture is non-uniformly distributed and made the measurement less accurate, which means if the background is uniform, you are going to see the measurement more and more accurate, but if the background is non-uniform, that actually is going to bring more and more trouble, because software does not know what is your intensity and what is your background. So, if it is a uniform background, it is actually going to give you the more uniform numbers, which means it is going to assign very comfortably the 0 to the every lower intensity pixel. But if it is a lower high, then it will not be able to judge where the your intensity of the spot is starting, where the your band is existing within the gel. So, that actually is going to make the measurements more and more erroneous. Many methods of background subtraction are possible. In one of the method, a replica image can be generated and then digitally subtract from the original image to correct the background. So, once you are done with the background corrections, then you can go to the next step and ask the software to give you the intensity. So, the next step is the measurement steps. So, once the lane and the bands are defined, it is possible to perform the quantification and the characterization steps. The amount in each band is quantified in comparison to the background information and the total intensity present in all pixels present in the band, which means if you have a band, you are actually going to get the intensity of each and every pixel of this band, and that is how, and that intensity will be in relation to the background what you have in the neighboring to the surrounding this particular protein band. So, if the known amount of a protein sample is loaded, then a calibration curve can be drawn and used to more accurately quantitate the protein band, which means if you have the known amount of the protein band, what you can do is you can measure the intensity versus the protein concentration, and you can be able to draw a calibration curve between this, and once you have that particular type of calibration curve, then you can be able to use this calibration curve to measure the intensity of the protein present in this particular band. So, this is a brief overview of the image analysis. Just to explain you these steps in more in detail, I will take you to my laboratory for an extensive demo on one of the softwares, where the students are going to show you each and every steps with the help of the softwares. These softwares are available from the company, so they may not be freely available to you for practice, but I hope this demo is going to be helpful for you to understand the whole procedure.
In this video, we will show you how to analyze a particular band of interest from SDS or agar gel electrophoresis gels using ImageLab software. So, here I will show you how to open the ImageLab software and how to analyze different components. So, you have to go to start and type ImageLab. So, it will open. You need not to do anything, just this is we are using new protocol. So, just have to go. First I will show you how to analyze the protein gels. So, this is our gel image. So, this is the molecular weight ladder, first row and last lane, and these are the fractions. So, we want to analyze how many different bands are there, first component, and what is the intensity of these bands and what is the molecular weight. So, I will try to show one by one. First, after getting the image, or you can get from you can open any ImageLab.scn file from instrument. So, go to here on left side top panel, you can see image tools. So, if you go there, there are different data. You don't want to see complete picture, so just you can crop the image and like this. Just say crop. So, this is the complete image, and go back here. After that, you have to identify how many bands are there, how many lanes are there. Just leave this to software, it will do automatically, or in another way you can select manually also. So let's see how it will perform. So click on lanes and bands. So lane finder, here lanes either allow the software to detect or you can do it manually. So if I ask for automatic, so it will see, you can see there are lanes given on the top of the gel, 1 2 3 4 5 6 up to 10. So this is something different, or if you want to do it manually also you can do. Suppose if you want to do enter number of lanes, how many lanes are there, so 10. So here you can adjust lanes. See, this is not fitting in the completely, so you have to adjust like this to get complete lanes. Now you can see the lanes are completely adjusted, fitted in the lane completely. So this is another way, manual way. So next, now after identifying the lanes, you have to go to bands. How many bands are there? So you allow the software to detect the bands. So here few options are there. Band detection sensitivity, one is low. Low means better for most prominent ones. So detection sensitivity low, if you are keeping it low sensitivity, then it will detect only prominent bands which the software can observe based on intensity. Otherwise you can do another thing, select high sensitivity means it will select a faint bands also, as you can see it has written there, or you can manually select sensitivity. So I am asking high, better for faint bands. See how it will do. See, these many bands are there, each and every single band it will be detected, although some of here are left out, but you can still manually add the band. So if you want only prominent bands, so in that case you just select low, which is better for prominent bands. So these many bands. So now question comes, you don't want to calculate molecular weight or intensity values, that is quantification for all these bands. Either you can keep these bands selected by software or you can remove. So for deleting, suppose if you want to add some particular band, suppose here I want to add this band, bottom of this lane, so just go add, just it added another extra band. So if you want to delete some bands, so you don't want these many bands, you want to only calculate for this prominent bands, you just go to delete, you just keep on doing till you get satisfactory bands. Okay, this way you can do. So you can add also. So after being done this, we have first detected the lanes, and in second we have detected the bands. Now it's time for calculation of or molecular weight analysis. So here the tricky part is you have to select the lane, which lane you have loaded the molecular weight marker. So I am selecting the first lane and the last lane. Okay, so it automatically came. So if you are using your gel of interest, So in that case, what you can do is, you need not to worry about, you just have to give your molecular weights. Suppose this is randomly taken, by default it has taken Biorad Precision Plus. You can change this one also, this pattern you can change also. Suppose you want to add some new protein molecular weight standards, you don't have this Biorad Precision Plus, you have loaded some other thing, but you know what are the first band appears on top to lower bottom band. So if you know, you can add new, just go new standard, or you can give the name. Suppose I am giving new molecular weight standards something XY. Okay, some company's name. So after that you have to add each and every band. Suppose first one I'm just giving these values only for convenience purpose, 250, and second band is 150. It need not to be these values, whatever your choice of marker is, you can give those values, those KD values. You need not to worry about what it has written, it will automatically disappears. And third band is just say 99, and fourth band is 78, and fifth band is 47, sixth band is 36, seventh band is 22, eighth band is 19, ninth band is 14, this is 12, and last one is you can keep 8. This is just for example purpose only, this is not actually any standard molecular weight marker, just for your understanding I am giving this one. After giving the molecular weights, you just click OK and you select OK. Yes, you want to, it is asking you want to apply. Yeah, yes, I want to apply. So see here, this is the molecular weight what we have selected. See here, new molecular weight marker XXY. So here you can see what we have given is, it is coming as it is. So the next thing is, you have to calculate molecular weight of these things. Okay, so you just need not to worry about anything, and you just go to analysis table. So here analysis table, it will give for each and every lane details with proper band percentage and lane percentage. Suppose in first lane it corresponds to first lane 1, it corresponds to molecular weight. See from lower weight higher one. So what are the values, different values, and absolute quantifier, this we will see in next part. And band percentage, how much percentage it is, these things we can see. So if you want to calculate, suppose in lane 2 you have lane 2. In lane 2, this is the fifth band I think. So it will give what are the molecular weight. So this is starting from bottom, band number, this is 1 to 5th band I think. So 5th band means on top, 5th band is 87.4 kD. This is 78, we are calculating for this one, this is almost comes 87.4 kDa, and the relative front is 0.187, uh, um, so in this way you can calculate molecular weight of the your band of interest. So this is all about molecular weight. You can save this thing and export this result to a new Excel sheet. So after being done this, now we are moving to quantity tools. So quantity means you know you have loaded something, what amount you have loaded in this lane. If you know that thing, we can calculate remaining lanes quantity, how much we have. So there is two parts. One is relative quantity, select a reference band on the gel. So you have to select one reference band. Based on that, it will calculate all the bands relative quantity, but it does not give any value. Suppose I am selecting this one, this is the reference one. So automatically it will calculate the remaining proteins, lane 3, this is lane 2. So it will calculate automatically relative quantity. It is taking 1 for this reference one. So based on this, it will calculate all the bands relative intensity. Suppose if you say this is 1, what could be this for band 1, it is 0.59, and band 2 0.15, 0.15, 0.06, 0.07. so in other lane 3 also, that band is, this is 5th band. So lane 3 band 5, that is molecular weight 7, the relative quantity is 0.89. so this is 1. If you are saying this is 1, this one is 0.89. so in this way you can calculate relative quantity. And you don't want this, you know some number, suppose how much protein it is present in this one. So in that case you can go to absolute quantity. At least you need 2 to 3 bands. So see, I am selecting this one. Okay, this is I am saying this is the quantity of 50 nanogram, and I am selecting this quantity of 65 nanogram, and I am selecting another one, this is 25. I mean automatically it gives. So you just take your mouse pointer to any band you want to calculate the quantity, actual quantity. So you calculate, you can click on that one, it will give automatically 72.214 nanograms. You want to calculate this band, so you just keep that one, it will give the value. You want to calculate this one, it will give the value. So in this way you can calculate absolute quantity of the any given band in the gel. So you can export these results, or you can save from this one also. So after quantification of the protein lanes and bands, you can use this for annotation also. So you can go to annotation tools. Suppose you want to indicate this band. Okay, you just give the arrow mark. Otherwise you want to show this band, so this is also another way. You can take this as screenshot, or you can save as a picture with these things, with annotation tools. You can add text also. Suppose you want to add text here, you just add something suppose lane 2, like that, lane 2 band 5. Okay, so this is the way how the analysis can be done using ImageLab software. So this is about all about proteins. So now I want to do the same analysis for the nucleic acids, suppose DNA. So just go so you have now you have DNA. So all the part is same as compared to proteins. So you can go to image tools for cropping. You can crop it, or whatever the part you want to keep, you can keep that one and say crop. After that, lanes and bands. So it is exactly same protocol for proteins and nucleic acid also. It will detect the bands, low better for prominent bands. I'm asking like that. Okay, so after that it detected lanes and bands also. Now you can go to molecular weight analysis. So here also, either you can give your choice of interest molecular weights you can give, and you can create new and add the values. This is here nucleic acids, molecular weight, base pairs. So in this way you can give, or you can use one that has given in already given in the software. So as you can see, this is already this is depicting here what is the base pairs amount of each lane. So here also this is same. So the next thing is, after being completed, you need not to specifically look into what this band corresponds to. You just go to lane 2 values. This is the lane 1, this is lane 2. Only single band we are predicting, that belongs to 78.6 base pair value. This is in lane 2. So as you can see, this is 80 and here it is coming, this is 78.6 base pairs. So in this way you can calculate the molecular weight of the nucleic acids also. And the other part is quantity tools. You can quantize the DNA also. So here also same in case of protein analysis. Here relative, so you have to select one band. Suppose I'm selecting this one. Based on this, it will calculate the all bands relative intensity, relative quantity. Or you can select a band, standard one, and this is quantity. This band quantity, I'm saying it is as 30 nanogram. So another band, at least we need to, this is as 10 nanogram. After giving two values, you can click any band. Suppose this one 39.92, this one 55, and this one 63, this one 64. So in this way you can calculate the intensity, absolute quantity of the any given band. You can use annotation tools also. by giving arrow or showing representing graphically also you can do same thing, and you can export the screenshot also. So you can save these things and you can export. Here the export options are there, you can do exporting also. So I hope this software information part help you to understand the how software can be utilized for analysis of different bands and molecular weight purpose or quantification purpose.
So, in this demo, the students have explained you how to select the lanes, how you can be able to measure the intensity of in every steps, and how you can be even be able to determine the molecular weight of the particular protein. Because if you have the markers, because in the example the student have shown that if you have a molecular weight marker run onto the gel on the in one of the lane, you can use that information even to calculate the molecular weight. So, with this we would like to conclude our lecture here. In the subsequent lecture, we are going to discuss about the some more aspects related to electrophoresis. Thank you.